The broadcast is now starting. All attendees are in listen-only mode.
Welcome back, everybody, to the third day. It's rather hectic for the organisers, so counting the days is the least of our worries at the moment. We've got a great schedule today, and we're looking forward to a whole host of different talks on a variety of subjects, mainly down to the amazing session we've got for you to start with. I won't say much more about that. A few things to note. I hope everyone's checking out the posters on the poster website. We also have started putting up videos of the previous sessions on that site. So if you have missed any talks, hopefully they're on there to check out. We had a second student mixer this morning. I assume went very well. And just remember, we've got one after the awards on Friday too. And just another reminder about the awards. There will be awards for any student, whether they're a member or not, for the best talk and the best poster and lightning talk. So there's two awards there. Hopefully the attendees are just creeping up, so people are still logging in, so we won't jump in too early. I found yesterday a fascinating day. I've got to admit, I actually made quite a few notes despite being under the cosh with emails and messages and sorting out speakers and all this behind the scenes. And for that, I really do have to thank Kimberly Bell, Alex Ball, and Vera Korosidis a lot for all their help behind the scenes so far. We had Jim help out with the chairing yesterday, and we've got Damien who's helping out from now on for the rest of the conference. So there's not much more to do for the welcome this morning because we've sort of covered everything. If there are any quick questions, do drop them in the question. If not, I will actually start early the next session, which is going to be chaired by Tim Riley, and I will let him introduce the session and why we've got the session. So if I invite Tim to join.
Sure, thanks, Steve. Hi, everybody, and glad to be here for the 53rd annual meeting of the AASP. As Steve said, we're here for a special session called Beyond Miscellaneous, honoring the legacy of Vaughn Bryant. Vaughn Bryant, for those of you who may not know, he passed away January 30th of this year, 2021, in College Station, and was really a big influence across a lot of subfields of palynology as well as beyond. You know, he basically had gone to University of Texas, went to University of Washington for a couple of years, came back and founded the Department of Anthropology at Texas A&M, and had his PhD actually in botany. And they asked if that was normal at the time, and he said, well, yes, of course it is. So of course it wasn't, and that's where the title comes from. The Beyond Miscellaneous, he had an article in American Antiquities in the 70s called Being Miscellaneous, basically saying all these academic departments want a ceramicist and a lithicist, but then they're wondering where all the people are to do Zoarc and PBOT and palynological studies in an archaeological context. Well, you're not hiring them because we're being considered miscellaneous as opposed to true, field archaeologists. And so we're now beyond miscellaneous, thanks to Vaughn. And so I want to just thank everybody for participating. Vaughn was a big influence on a lot of us in lithopalynology and forensic palynology in the Americas and a number of other fields. And I really appreciate and was impressed to see so many people from across his entire career step up to do these presentations. And with that, I'm going to turn it over to Dr. Jim Adovazio with his presentation on the pollen from Meadowcroft Rock Shelter, Washington County, Pennsylvania, an important early site in the Americas.
There should be a little red microphone button on the panel on your screen, which you'll have to click to unmute yourself. There you go.
Okay, am I on now?
Yes, yeah, we can hear you and we can see your screen, but right now we just see the desktop.
Okay.
So if you could bring up your presentation.
How exactly does one bring up the presentation? I'm not altogether sure.
It's in PowerPoint. If you open the file in your PowerPoint.
All right, one moment.
There we go.
That's it.
And then if you put it into presentation mode, exactly. Yeah, perfect. So we can see your presentation and we can hear you. Please proceed.
Okay. Meadowcroft Rock Shelter is a deeply stratified, closed archaeological site located on the unglaciated Allegheny Plateau, 46 kilometers southwest of Pittsburgh in Washington County, Pennsylvania. Those are various views of the site as I originally saw it in the spring of 1973. And the view on the extreme left is taken from the south bank of Cross Creek, looking up at the rock shelter, which is in the center of the image. The vegetation, which is shown, marks the current drip line of the site. Principally excavated with extreme care between 1973 and 1978, with subsequent excavations intermittently in the 1980s, 90s, and more recently. And that's the 1973 excavation before the addition of the roof, which covers the site. This is one of the more recent excavations done in the 1990s. Meadowcroft was, by all accounts, the first archaeological locus to seriously challenge the then prevailing paradigm, which stated that the first inhabitants of the New World were the manufacturers of the iconic Clovis fluted projectile points. Additionally, these were thought to be rapidly moving, highly specialized big game hunters who played a prominent role in the extinction of all of the Pleistocene megafauna. Early in the excavations, but well after Meadowcroft had generated acrimonious debate and extensive criticism, I, who was the principal investigator, approached Vaughn Bryant to ascertain his interest in possibly extracting and analyzing pollen from the sediments of the site. Vaughn was pointedly warned that his tentative connection with this highly controversial locality may not be the most appropriate choice for his then nascent career trajectory. I know this will be alluded to later in the presentation. Vaughn indicated that despite his misgivings about pollen preservation at this locality, as well as the often vitriolic criticisms of the site, he would provide an analysis which he duly produced on 4 December 1975. The results of this analysis were never published, and it is from his report that the following comments are largely abstracted. The 45 sediment samples from Meadowcroft were personally collected by the principal investigator, who literally sampled all the Holocene sediments. And here you see the 16 feet of Holocene sediments at the site, with the tags marking various places where geological columns were extracted from the site or where archaeological features are located. You can see on the second step the cross-section of rock, which shows how they were removed from the site by drilling and piecemeal. The samples were collected from continuous columns, both inside, which is what you have there, and outside the modern drip line. This is the area from outside the current drip line, which shows where the columns were extracted that Vaughan ultimately analyzed. The procedures Vaughan used to chemically treat each of these sediment samples incorporated all of the then approved laboratory protocols, which I don't need to go over with all of you who are palynologists. Essentially, these procedures consisted of a series of steps that all of you are intimately familiar with. I'll go directly to the results. As initially feared and as addressed more shortly, pollen preservation of Meadowcroft rock shelter was extremely poor. An examination of each sample showed that most contained at least some charcoal and a few fungal spores. However, only a few of the 45 samples contained any pollen at all. When pollen was present in these samples, it was poorly preserved and was sparse. No samples contained sufficient pollen to actually conduct a serious analytical study. The contents of each analyzed sample are listed in these tables, which follow. The lack of preserved pollen could have resulted from any number of causes, including microbial destruction and oxidation. Certain groups of fungi, such as the Phycomycetes, feed upon the cytoplasm found inside pollen grains. In order to reach the cytoplasm, the fungal mycella produce enzymes which are able to dissolve portions of the hard outer walls of pollen grains and thus gain entrance. This process severely weakens the pollen grain and enables other destructive processes such as oxidation and mechanical weathering to destroy the pollen grain before it can become preserved as part of the fossil record. Again, you're all familiar with that phenomenon. Studies have shown that bacteria also play a destructive role in preventing pollen grains from becoming preserved. Certain types of bacteria, like certain fungi, also feed upon the internal cytoplasm of pollen grains and are able to enter the grains by penetrating through the hard outer wall called the exine. It is difficult to determine the presence of these fungi and bacteria in ancient sediments, and even more difficult to determine whether or not they played an active role in pollen destruction. This is the table, the second table, which shows the results of the analysis outside the contemporary drip line. Additionally, repeated wetting and drying of sediments can often speed the oxidation of non-carbonized organic material, including pollen. Since at least some of the sediment samples at Meadowcroft rock shelter were known to have undergone repeated wetting and drying, especially those shown in this table, Brian assumed that this form of oxidation played an important role in pollen destruction at this site. Finally, the oxidation of pollen can also occur when pollen comes in contact with fire or intense heat in the presence of oxygen. Since charcoal flecks, as this table and the preceding table indicate, were a common component of most sediment samples at Meadowcroft, Vaughan assumed that fire and heat were also important factors which led to the near total loss of fossil pollen at this site. For all or at least some of the reasons specified above, pollen was very scarce at Meadowcroft rock shelter. Nonetheless, it is perhaps significant that all of the identified pollen grains duplicate plant species identified by one of Vaughan's early students, Kathleen Cushman, in the very large plant macrofossil collection from this locality. Incidentally, Cushman's macrofossil identifications were confirmed by Von Bryant personally. They numbered some 1.4 million specimens, and it took literally years to analyze all of them. All of this in turn suggests that the still prevailing interpretation of the floral sequence at Meadowcroft Rock Shelter, which you see an image of here and in the daytime and at night, is correct. The interpretation stresses the basic continuity of plant communities throughout the Holocene at Meadowcroft, albeit with minor fluctuations in species frequency. Since the completion of Vaughn's analysis, this interpretation was extended back into the late Pleistocene. As all of you are perfectly aware, this was one of the reasons the site was heavily criticized initially, was because of the failure of the critics to note the lower elevation of the site and the attendant changes to the biotic communities in this area occasioned by that. Since all previous pollen work had been done in the uplands of the Allegheny Plateau, the difference between the pollen analysis at Meadowcroft and the plant macrofossil analysis at Meadowcroft, as well as that on the upper Allegheny Plateau, was striking. That essentially summarizes the results of the pollen analysis. If you have any questions, I'd be glad to try and answer them.
Sure. I have a question for you. So how did it feel after, basically almost 50 years after the discovery that Meadowcroft is pretty well accepted now as a early habitational site in the Americas with some of the radiocarbon corrections and things like that?
I think I drastically, as did Tom Dillahay at Monteverde, underestimated how long it took paradigms to fundamentally change. I think all of you here in this session are familiar with paradigms in your own area and how rapidly or slowly they might change. So I never imagined it would take 5 decades for the perspective to alter, even when a lot of European archaeologists said that the scenario that was presented Clovis first, so to speak, was absurd from the very beginning. And the South American archaeologists said we have material here as old as Clovis, but clearly is of a different type and represents a different technology. The US and Canadian responses to this were typical American responses. Those guys on the other side of the Atlantic have absolutely no idea what they're talking about, and those guys in South America don't even speak English. As a consequence, it has taken literally a very long time for this change to occur.
Right, and we'll be hearing about some other pre-Clovis sites here a little bit later where pollen has played an important role, along with some of the other studies Vaughan really kept alive in the Americas, like coprolite studies. So I think this dovetails pretty nicely.
Well, thank you very kindly.
Yeah,
I was delighted to participate because, as I indicated earlier, and as will be repeated later on in the summary of this session, when I approached Vaughan, this was the very first project he did outside of Texas. And I did duly warn him that any connection with Meadowcroft at that time was potentially hazardous to his career health. But he nonetheless persisted, and persisted to his credit, I think. And I'm delighted that the results ultimately have been accepted, and especially his contributions thereunto.
Yeah, Vaughan was never one to shy away from controversy, whether it's helping along with some Bigfoot tracking up in Washington or almost anything else. And so that's one thing that's really benefited him and a lot of his students and throughout the years.
Well, again, it was a pleasure being here.
We certainly appreciate it. And with that, we'll move along, assuming there's no more questions. I'll talk slowly here for a second. We're going to move to another one of Vaughn's early students, not the quite earliest, but pretty close to it, Carl Reinhardt, who's going to talk about coprolites and the ancient gut microbiome, and some of the interesting things that started with pollen and moved into all sorts of other microfossils and evidence of past diet and health.
All right, can everybody hear me?
Yeah, we can hear you just fine, Carl.
And can you see my screen?
Yes, we can see you.
Yes, sir.
Fine.
Okay, so what I'm going to present is some data related to this Nature article that came out a couple of months ago, designed to recover an ancient gut microbiome from a number of coprolites. I began doing this work in February of 2019, and every step of the way, I shared the information with Vaughn and was able to lecture his classes at one point in late 2019, and was happy to share the galley proofs. So I'd like to dedicate this presentation to Vaughn, who was unfortunately not able to see the final publication, but certainly the galley proofs he was interested in. Now, my participation in this research was to design or develop assessments for preservation potential, determine the biological source, whether dog or human, for the coprolites, and verify geographic origins of some of the coprolites excavated by the Peabody in the 1930s. That issue is a bit of a problem because several different excavations were done by the Peabody in the 1930s, including in the Great Basin and also in the Colorado Plateau. So I do the analysis of the background pollen to verify that the coprolites in the 30s were from the Colorado Plateau. We selected pinworm, aDNA, and eggs as a method to determine human origin because we had two chances of getting at the pinworm, which is a human-specific parasite, with the genetic analysis as well as the analysis for eggs. Problem with this analysis is all the samples were between 0.1 and 0.9 grams, which is a lot less than what I prefer to work with is 3 grams to 5 grams. So I really focused on microfossils, used lycopodium quantification, and used SEM, scanning electron microscope, for most of these criteria. The preservation was what I did first, so that I screened the coprolites that were sent to my lab and then assessed whether or not they had good preservation, bad preservation, or excellent preservation. One of the conclusions from the gut microbiome study is that we had hit upon, or they had hit upon, an ancient gut microbiome that was largely extinct. Surprisingly, ancient feces had over 400,000 different bacterial genes. And by comparison, feces from developing countries, indigenous countries in Mexico, had 300,000. Industrialized folks, such as ourselves, have 200,000. So what we see in this study is a loss of bacterial genomes and bacterial species, and most important, enzymes. And those enzymes focused on digestion of chitin, cellulose, and other types of materials. For the future, we're working towards defining this extinct gut microbiome beyond Mexico and Utah, Durango and Utah. What we want to do is several different things. We want to compare specific genes, specific species, with the macronutrient composition of the diet, and especially look at fiber sources related to specific genes and enzymes that are discovered in the coprolites. And to that end, we're looking for coprolites from all parts of the world. We're having some difficult trouble finding coprolites in the old world. So if you know of samples from the old world, hit me at either of those two emails. Finally, and perhaps the most wishful aspect of this work, is we want to create, well, the microbiome folks want to reproduce in a Triassic park sort of way, ancient human gut microbiome to be used eventually as intervention to deal with modern health problems. Jean Blanc edited a couple volumes dedicated to coprolites and archaeological and anthropological sciences. I did a paper with my students on imaging coprolite taphonomy and preservation. In that work, we identified 4 different categories of conditions that affect preservation of coprolites. What I determined as best in reviewing all these data was that if the coprolites came from dispersed middens or refuse which incorporated a lot of vegetal remains like sandals, cordage, quids, etc., we had good preservation. If these samples came from deep caves or rock shelters far from the drip line, we had really good preservation. If we had no infiltration of dry substrate and no fluid infiltration of calcium, then we were in good shape as well. Finally, if we saw no decomposers associated with moisture like fly maggots, mites, nematodes, and fungus, that hinted at good preservation. Oddly enough, the dry decomposers, spider beetles, seem to not affect preservation of microremains and also the gut microbiome. EV in this table stands for pinworm. The scientific name is Enterobes formicularis. Because of the small sample sizes, I was not able to identify pinworm eggs for Boomerang Shelter or for the Peabody, Colorado Plateau, Utah site. However, the preservation, as following my procedures as I summarized in the previous slide, was excellent. They were all agricultural sites, fairly recent in history, and I have to move this so I can see. Oh, the microbiome preservation for those first 2 sites was excellent, and even pinworm DNA was present, so we could verify for the first 3 sites we were studying that we had excellent gut microbiome, we had excellent preservation, and those 6 samples, the 6 samples from those sites are the basis for the data that was presented in this paper. Essentially, I screened 66 samples. Of those, 6 had well-preserved microbiome DNA. The Dyke cliff dwelling in Verde Valley, we ran out of money. One of my students is doing his thesis on the standard coprolite analysis, but we need another grant to continue with that. The other sites that had good or poor preservation had no microbiome bacterial preservation. Okay, palynology has contributed to a long time to identifying fiber. And I think if we look back to Martin and Sherrock's work in 1964, they identified cleome as a hallmark with corn, beans, and squash of ancestral Pueblo diet. And I think every coprolite analyst involved in pollen since that time has verified that a source of fiber in ancestral Pueblo diet is cleome. This is the kind of information that the genomic folks want to focus on to determine whether or not there are specific genes and species that are focused on specific fiber sources. Another thing that came out in this paper related to corn fungus. We found, I found a lot of Ustalegomatus spores, which is Huitzilopochtli in Mexico and Nanya and Hopi. We call it corn smut in North America. And people ate this fairly commonly from Durango, Mexico, up to Utah in agricultural times and up into historic times. What was found by the gut microbiome folks is chitinases associated with these spores. Up to 9 billion spores can be found per gram of coprolite. And Batillo, in her 2018 paper, really defined the role of this fungus in ancestral Pueblo diet. And it had a large time and space distribution. It was still a modern Pueblo, Tarahumara, and general Mexican food as well. I used a scanning electron microscope to verify that we were really looking at Ustilago metaspores, and we are, and also to verify that this incredible number, incredible to my mind, is over 900,000 spore items per gram. And as I mentioned, the highest concentration I found was 9 billion spores per gram. What we're looking at the right photograph is just a smear of screened coprolite residue, and most, if not, nearly all of the identifiable structures in that coprolite, as we see in this view at the SEM, are Ustilago metaspores, big time food. Now, the other thing that we want to do in the future is look at fiber origins. So we've been discussing in Zoom meetings prior to setting up the next grant proposal as how to identify fiber sources. Certainly, trichomes are great. At Aztec ruins, I've identified phaseolus leaves, bean leaves, as a major dietary source based on trichomes. And in a scattering of coprolites from the Colorado Plateau, I've also identified Descurania based on those trichomes, which is tansy mustard. But I do believe pollen can be used as a proxy gauge of fiber sources with some caveats. We want to see that for Anemophilus pollen, I have a cutoff of 20,000 or more grains per gram with clusters before considering that they might be dietary in origin. And also I want to assess the condition, which I'll show in a couple of slides. For Entomophilus types, I want to see more than 1,000 grains per gram, again with clusters. And finally, for ZMAs, and this cutoff may seem extreme, but I want to see 100 grains per gram to interpret whether we have ZMAs as evidence of diet. And that comes from discussions with archaeologists at Aztec ruins and Solomon ruins, who pointed out that ZMA, corn pollen, is a big background contaminant within pueblos. When I said assessment, what I want to do with the scanning electron microscope is look at the preservation of the pollen. So on the right, we see a quinoam grain. It's folded. All of the opercula are missing on the pores. Whereas in contrast, the grain on the left shows a little bit of compression, but overall the pollen grain is in good shape. All of the opercula are present. So when I see pollen like this in a coprolite, I feel confident, especially when we have greater than 20,000 grains per gram in clusters, that I am looking at a dietary source of that pollen. Sarcobatus was kind of interesting in this analysis of the coprolites from the Colorado Plateau, basket maker times. I found 2 samples that had the male cones, the male flowers in the macroscopic remains. The microscopic remains revealed 73,000 and 87,000 pollen grains per gram of Sarcobatus. Again, looking at these grains with the scanning electron microscope, I see the opercula still on the pores of the grain, which suggests to me all of this is good for an interpretation of a dietary origin. One of the things that has perplexed me for some years is this phenomenon of what I call ballooning corn pollen. And when I see concentrations of ZMAs over 1 million grains per gram, I see a strange alteration of the pollen wall. The size of the grains expands 2 to 3 times, and then the thinned X sign bursts. So these look like inflated and then burst balloons. And I think that we've been considering that the ancestral Puebloans and other archaeological folks collected and ate maize pollen. What I'm very interested in is collaborating with my gut microbiome colleagues to see if we can identify genes, enzymes, and genomes that occur with pollen consumption. And so that is my major goal, to give you an idea of what I'm talking about. This is a picture that shows a normal ZMA's pollen grain. Down here, I'm gonna move this panel over there. Okay, normal pollen grain, and then here with the arrow, we're looking at an expanded pollen grain. The arrow points to the aperture, which is way bigger than what we would anticipate for a normal pollen grain. And then these other structures are sheets of burst pollen grains. And incidentally, you can see Ustilago or corn smut spores along with this pollen. going up close and personal, here is 6 pollen grains that are burst and are represented only by exine sheets. And the arrows point to discernible pores on the surface of these grains. And essentially, what this shows is that the exines thin, split, and even when they're overlapped 1 or 2 over the original levels of pollen, you can still make out those pores, which helps with determining egg per gram. Going close up and personally, what I did is counted the number of scabri per 25 square micrometer areas. A shows a pretty standard exine of a uninflated maize grain. And in that, looking at a series of those pollen grains, I found there are about 148 on the average scabri per 25 micrometers squared. As we go through B, C, D, and E, those are pollen grains that are more extensively and severely expanded, ballooned. And one can see that the exine is actually stretching. And as the exine stretches, the numbers of scabri or spinules per 25 square micrometers decreases to 45, 33, 32, and 20. Interestingly, along with this decrease in the number of scabri, we can see a more clear definition or resolution of the micropores on the surface of the pollen grain. So you can actually start making out those pores as a pollen grain expands. Now, so that is a brief summary of some of the highlights of the work that came out of this collaboration with my microbiological and genomic colleagues. And I really appreciate Von's input. When I first started finding thousands or millions of pollen grains per gram, Von asked, how many angels can dance on the head of the pin? And I think he was asking me to think about quantification more thoroughly. However, all I could imagine was very tiny celestial figures, each one carrying a pollen grain. So that didn't work for me. However, I think he eventually agreed that I am looking at some very interesting dimensions with regard to maize pollen. All right, now I'm going to try to share my screen. If I can do that, I don't think I'll be able to figure that out. Are there any questions? Did anybody hear me?
Yeah, we heard you, Carl. That was actually really great. And I certainly appreciate. Oh, there you go. There's your face.
Yay,
Yeah. As you say, sort of the dietary role of pollen. It's been a long question with some of the stuff up in the Fremont area where I am, and what does quantification really mean? And I know you've gone back and forth with some of Vaughn's other students in the past, in Society and Journal of Ethnobiology and other places regarding that. So it's really cool to see it's still being part and being brought into the realm with all the molecular studies and the SEM studies, and really fascinating seeing those exploded corn pollen. So, and I'll shut up in case if somebody else has something they want to say.
There haven't been any questions coming on the question message boards yet. Please, if people do have questions, they can use the question function on the dashboards to ask questions, and I'll ask them at the end of these talks.
Right, you're certainly welcome to do that, or if there's time, like there's right now, we got a minute or two. You're welcome to.
Bryles. I don't know if you can hear me, Carl.
Hi.
Hi. So I'm interested in the number. So the clustering of pollen that indicates use, right? How many grains do you say, or do you look for before you say, okay, this is definitely an indication of use?
I look in essence for clusters of greater than 20 grains.
Okay.
But I'm more comfortable not only with the number of grains per cluster, but how many clusters are in the count overall. So I count each cluster as a single pollen grain. Sometimes I can make out like 20, but when it gets beyond 20, the clusters became too thick to really count the actual numbers of pollen grains there. So I focus for anemophilous pollen on 20 grains. For anemophilous and maize, I'll talk about maize first. Oftentimes with maize, the clusters occur in many linear patterns or lines of pollen grains. For the anemophilous pollen, clusters associated with fibers are important as far as I'm concerned. And one of the things I've done with my work at Aztec Ruins is with squash pollen, use acetylosis very, very sparingly in order to maintain some of the fiber connections from the male flowers.
Oh,
So,
interesting.
from my opinion, it's not just the clusters. It is how the pollen is arranged in those clusters. And then if I can identify fibers inherent in that cluster of pollen.
Awesome. Thank you. It's a good, really great. Great. That's awesome. Thank you. Been wondering that for a while.
Okay.
Well, great. And with that, we'll move on to already mentioned today, John Blong, who did edit some really great volumes on coprolites recently in Archaeological and Anthropological Sciences 2019. And I'll just tell a brief story. The first time I was doing coprolite research for my dissertation, and John was a student at the Center for the Study of the First Americans at Texas A&M. And I was rehydrating and screening coprolites for macrobotanicals while he came in to take this class that I was a lab TA on paleoethnobotany, and had an adverse reaction to the very inherent physical nature of coprolite processing. And so I love the fact that John is now not just, partially a coprolite person, but really it's one of his main specialties. So with that, I'll turn it over to John Blong, who's going to talk to us about the early to middle Holocene subsistence in the Great Basin, specifically the coprolite evidence from the Paisley Caves in Oregon.
Thank you, Tim. I'm glad you told that story. I was actually going to tell that too, but just to clarify, it was Tim had rehydrated a coprolite and was pressing it through the screen, and I had this involuntary gag reflex when I saw him doing that. And so, but yeah, as Tim said, here I am now many years later, and I'm studying coprolites myself. So, okay, I'm going to share my screen here. Okay, so I'm just looking at the structures here. Are you all looking at my screen, or am I just looking at my video here?
So far, we can just see you.
Okay. Sorry about this. It says I need to grant security access here.
All right. It does show that you have screen control in the little panel. So.
John, did you get a little box pop up that asked you to share your screen?
Yes. Yes, and it says it's asking me. It says I need to grant access. Sorry about this. I'm holding things up here. It says I need to grant access, and for some reason I can't seem to grant access to it.
Blessings and curses of these online meetings, right? So we can meet from all over the world, but it's always a little touch and go.
Yeah. Well, I can start talking, and then maybe I can try to figure this out. or I don't know if this is going to be distracting for me. I'm really sorry. I'm not quite sure what, I mean, I'm on a Mac computer and I'm not sure if that's what the problem is here, but anybody have any suggestions for me?
I'm wondering whether you could send your presentation to somebody who could share their screen, like myself or Tim, and just ask us to pass the slides on. So it sounds as though it's your system.
Yeah.
I've had Macs before and that works, so I'm not sure what. So I don't know if you want to do that, and we can maybe move on to the next speaker if they're ready and do it that way, or it's up to Tim. Do you have any suggestions like that?
No, I think that's a good one. We're happy if let's see, we've got yeah, we got Angie up next, and I think I saw she was on here. We could do that and just flip them.
I'm ready.
Okay, and then why don't you send that? Yeah, if you have Steve's email, that's great. If not, you can send it to me, and I should be able to. I'm actually on vacation at my folks' house, so using a computer I'm not used to either. So Steve's probably the best option. If you just want to forward the slides to him, and we'll flip the presentations. Does that work?
It works for me.
Great. Okay, well, very good. So we're going to come back to John Blong's talk on coprolites in the Great Basin, and we're going to move on to Angie Parodi's diversity in vegetation and bioresponses after Pleistocene mega herbivore extinction events across the U.S. Excuse my Eastern U.S. Excuse my butchery there, Angie. I know you'll do a great job introducing it.
Yeah, I was gonna say I'm ready if I can figure out how to share my screen. Okay, I think I've got it. Okay, there we go. Okay, so I'm sharing my screen right now, right?
We can only see your video and hear you.
Okay. Sorry. Okay, sorry, just a second. Here we go.
Yeah.
It is asking me to quit and reopen, so I might pop off of GoToMeeting for a second.
Okay, we'll be here. Well, folks, sorry about all this. As I said earlier, the blessings and curses of online meetings. We get to meet from anywhere in the world, really, that has a decent internet, but sometimes it doesn't go smoothly. But I didn't really prepare any fill-in stuff. If anybody has any great stories they want to tell of Vaughn while we're waiting for Angie or John to pop back on, I'd be happy to hear them. I know we're going to have some great stories that Chris Sobolik has compiled here in a little bit, but there's John.
Yeah, so I'm back, but now the option to share my screen has gone away again.
Oh, it's telling me you are off. Oh, there's two of you now. So it might take one of the organizers to share it with the new Angelina. Right, because it does show me you have, oh, there you go.
Okay, I should have cleaned up my sticky notes over here on the corner. I'm quite embarrassed at my shopping list being projected for everyone to see.
We can see your screen and we can hear you.
Okay, good. Yeah, I'll go ahead and get started. So I did my PhD with Vaughn in 2018, and then now my postdoc is in geography and geosciences. So I really kind of embody this idea of being miscellaneous and not really fitting into a single box. So that's what my research will be showcasing today, is just the challenges and the benefits of trying to put so many different disciplines together into a single research talk. So today I'll be talking about diversity in vegetation and fire responses after Pleistocene mega herbivore extinction across the eastern US. Really quickly, I just wanted to acknowledge my collaborators and co-authors here and the 2 National Science Foundation grants that this work was funded by. So, climate change is changing landscapes before our eyes. We know this with fire, increasing droughts, increasing heat in the recent IPCC report to come out, but humans have more influence than just climate. So, for example, drastic differences can exist here between just 2 pastures, like shown on the left, where divided simply by a fence, overgrazed pastures on one side are pushed over a tipping point when so much native vegetation is lost that the natural prairie sod, which keeps away invasive species, can never recover. And alternatively, the complete removal of herbivores from a landscape can result in woody vegetation encroachment and a loss of grassland habitat, such as this example here by Middleton 2012. So the prairies of North America are relic ecosystems, which were once maintained by herds of large herbivores. And these herbivores, which I'll be calling mega herbivores, defined as having a body weight over 100 kilograms, include animals such as mammoths and mastodons. So a lot of these changes that are happening in the present are happening in real time, and it's really difficult to pinpoint the relative influences of herbivore behavior, climate change, and other influences such as fire suppression. So the Pleistocene epoch, shown here in this middle panel, spanning about 2.5 million years to 11,700 years ago, provides a laboratory for investigating these questions of rapid climate change, increasing human influence, and a mass extinction event, which included nearly all large herbivores in North and South America. So this talk is going to focus specifically on the last glaciation in North America from about 20,000 to 8,000 years ago. Really quickly, just to remind people of some terms here, we already talked about Pleistocene and then the Holocene directly following the Pleistocene. I'll also be discussing a couple of climatic events, the Bowling Allerod from 14,700 to 12,900. BP, which was characterized by very warm temperatures, and then the Younger Dryas, which was characterized by a sort of return to glacial conditions. 2 other significant events that occurred during this time are human arrival by 15,000 BP at the latest, and then megafaunal extinction by 12,000 BP at the latest. So despite widespread and interdisciplinary focus on the causes of Pleistocene mega herbivore extinction, my talk today is going to focus primarily on the effects of this extinction. So thinking back to the beginning of this talk, recall the influence of the large browsers and grazers can have on a landscape. For example, this work by Jacqueline Gill indicates that vegetation and fire change follow mega herbivore extinction. So here you can see Sporamiella decline, Sporamiella being a coprophilous fungus, which is often used as a proxy for large herbivore abundance, which is followed by an increase in no analog pollen assemblages, reflecting plant communities which have no modern analog, an increase in pollen from hardwood trees, and then finally an increase in charcoal, which represents an increase in fire frequency and intensity. So here I'm going to present a synthesis of 14 coprophilous spore records from Eastern North America in a broad transect from the north being the Great Lakes region and New York State down to Peninsular Florida. This synthesis combines new and previously published data and represents over a decade of field and lab work. And the cornerstone of this synthesis is 5 new recounted spore records indicated in blue here, which unfortunately I see now that there's a couple of extra blue circles. So here represent the 5 recounted spore records. Specifically, I'm combining the Coprophilus fungal spore data with existing pollen and charcoal records from each site to investigate the influence of megaherbivory release on vegetation and fire. I assessed the timing and the strength of Coprophilus spore declines across the eastern US. And I also am comparing those to the evidence brought to us by direct bone dates. I'm then evaluating the megaherbivore release hypothesis by assessing associations among spore declines, increases in deciduous hardwood pollen, abundances of no analog vegetation, and shifts in fire regime. So original pollen residues were used on the newly recounted spore records. I aggregated the data by using 10 strongly coprophilous fungi records. The spores in this talk are shown as a percentage of the total pollen. Oftentimes, spore concentration or accumulation is preferred, but due to invalid tracer counts in several of the samples, we had to use spores as a percentage of total pollen. And then I also aggregated the total hardwood pollen counts into percent. And you can see here, one of those strongly coprophilous fungal taxa here is spore miela on the right. Charcoal accumulation data was analyzed using char analysis. No analog vegetation assemblages were evaluated using the minimum square core distance metric. All age depth models were reconstructed from each site, recalibrating the dates using the new INCAL 20 radiocarbon curve, and then modeling the age depth using the program R-Bacon. Bayesian change point detection analysis was utilized to determine major changes in the means of fungal spore abundance at each site. I used the BCP package in R, which calculates the posterior probability of an abrupt change in pollinomorph abundance. So here you can see your standard stratigraphic pollen diagram with older on the bottom and younger on top. And now these darker blue polygons are the spore percentage as a total of the pollen abundance. And then on the left here is the BCP change point analysis, which shows the probability of a significant change point in that record. So really, you can see here divided by regions that there's no clear spatial trend in the timing of spore declines, with both the earliest declines and the latest declines occurring within the southeast here. But I've highlighted the area here between roughly 16,000 and 13,000 BP, which show the decline of most sites in this record. Here, this information is summarized in a table, so you can see the regions on the left, the sites in the middle, and the ranges on the right. So note that there are ranges because of both depth uncertainty and also age modeling uncertainty. The gray boxes indicate that the signal is weak due to low overall counts or inconsistency among the different spore quantification methods. But the timing of these spore declines occur roughly between 16 and 13,000 years, and this falls generally between the window of earliest human arrival and the last known dates of megafaunal extinction, which suggests that perhaps spores are recording an ecological signal that was a precursor to the final extinction. Our best available data here about megaherbivore extinction occur from some recent modeling projects. So here in the Great Lakes, Widja et al. Estimated an extinction window of about 13.5 to 12.6 thousand for mammoths and 12.8 to 12.5 for mastodon in this Great Lakes region. This figure demonstrates the discrepancy between coprophilous fungal spore declines and these final bone dates, where spore declines are obviously occurring much earlier. So Gill et al. Have hypothesized that this spore decline indicates a functional extinction, where mega herbivore populations are low enough that they have very little impact on their ecosystems, and that this decline actually predates the actual extinction. However, this has been challenged because you can see here that it looks like there's an increase in faunal remains right before extinction, which indicates potentially a boom-bust cycle, but it's also possible that there is an increased prevalence of wetland conditions, which are suitable for bone preservation at this time. Similar research exists in the Northeast US, where once again, a spore decline predates extinction estimates for mammoth and mastodon. And Widja et al. Recently compiled Pleistocene faunal dates from across the southeastern US. However, the faunal record in the southeastern US is sparse due to a lack of stratified, well-preserved sites, and most bones are depleted of datable collagen. So many of these dates are on bioapatite, which is generally considered to be unreliable. However, it still appears that many mega herbivores survived into the late Pleistocene, and in many instances after the spore decline. Archaeological context can help us potentially reconcile the fungi and bones here. So human and megafauna overlap for millennia, but early spore declines do roughly coincide with some of this earliest evidence of human migration to North America. So some potential anthropogenic influences that may have caused a spore decline before the bone decline include this idea that perhaps humans were actually caching hunted animals in bodies of water in the Great Lakes region, in these kettle ponds, acting as sort of a cooler that would help preserve the meat longer. In the southeastern US, it's possible that if the Younger Dryas was relatively dry, lakes could have been monopolized by early humans, which would drive mega herbivores away, which would result in fewer spores accumulating into the lake. And now this is a big table with lots of numbers, but the main thing to note is just these colors. This is building on the table I showed you earlier with the spore declines. Here we have a color-coded table that show which sites and which signals support the mega herbivore release hypothesis. So light blue being support of mega herbivore release hypothesis, yellow being potential support because it often slightly precedes or co-occurs with spores declining, and then red being that it does not support the mega herbivore release hypothesis. So here, the main thing to take away is that you can see that in the north, the mega herbivore release hypothesis is generally supported, and in the southeast, it is not well supported. So these regional vegetation and fire responses to mega herbivore decline include the northern US, where we have an increase in hardwood and no analog vegetation and fire activity. The upper southeast US, we have an increase in hardwood vegetation, but no change in fire activity. In the lower southeast US, there's a decrease in hardwood vegetation and an increase in fire activity. So this is, could be an entire 60-minute talk just kind of walking through the potential reasons that we see such a different response in the different regions to mega herbivore decline. But here I'm just going to mention this hypothesis that the Southeast is a warm thermal enclave. And this is evidenced by a lack of mountain glaciers in the Appalachians, despite the location of just 500 kilometers south of the southern ice sheet extent. However, and also geochemical temperature indicators suggest that the southeastern US was relatively warm, particularly peninsular Florida actually warmed during the Younger Dryas. So it's possible that this change between the southeastern US and the northern US in the response to mega herbivore decline could be reflecting some sort of climatic signature as well, where maybe the drier climates, the drier, colder climates of the north, where biodiversity, or sorry, where vegetation productivity is lower, perhaps there we're seeing that they're more vulnerable to mega herbivore decline. And finally, this wouldn't be a talk honoring Von Bryant if we didn't consider, what this proxy actually means. So we have to consider how we're representing these spores. It's currently, there are many different ways that people are looking at these spores, whether it's concentration versus accumulation versus percent of total pollen. And each of these methods is vulnerable to different factors. There are also different environmental preferences of fungi that we need to consider, as well as basin size, shape, and coring location. And finally, conclusions and lessons learned from Vaughan. Dung fungi do show a consistent abundance and decline in the Pleistocene eastern US, although this is consistently before bone dates. So more research is needed to understand what that means. Is it a functional extinction? Is it a taphonomic problem? That mega herbivore release hypothesis is supported in the northeastern US and it is unsupported in the southeastern US. And then finally, that the taphonomic considerations are important, and this proxy is still under development. So one thing I've always appreciated about Vaughan is that he had the ability to be a big picture thinker and to answer these big questions, but he was always trying to work to make these incremental advances in how we understand the formation of the pollen record and our assumptions and how we are and how we are extracting and recording these signals through pollen. So I've always appreciated that. And here are just a few good pictures. This one over here on the right is my favorite because I got to visit him at MD Anderson when he was in isolation for 6 weeks, and he convinced all of the doctors to let him have his microscope. So I got to bring him his microscope, and the doctors were certainly, that was the first for them. They'd never had a patient request a microscope in the isolation ward. So I'm not sure if I have any time for questions. I think I probably don't, but thank you all for inviting me. Thank you for Tim for organizing this session.
Angie, great. We really enjoyed that talk. Yeah, we are running a bit long for those of you who are joining us. We had a couple little technical glitches and we switched Angie with John Blong. So I'm going to turn it over to John Blong, and he's going to tell us all about coprolites in the Great Basin.
Okay, I think there we go. So actually, I do think I figured out my my screen issue here. But are we all?
Yeah, we can see your screen, we can see you, and we can hear you. So we're good to go.
Okay, great. Just need to figure out how to advance my slides now.
So John, I think currently Steve is sharing your presentation. Did you want to try and share it yourself, or do you just want to tell him to advance when you need to advance?
We can just go with Steve. So that way we're not holding up anymore, waiting for me to figure this all out. Okay, thanks everybody. And again, I apologize for the technical difficulties. So yeah, the work I'm going to present today actually kind of builds off nicely from what Carl was doing there, what Carl was talking about. And I'm going to talk today about some of the work we've been doing at the Paisley Caves in Oregon with a multi-proxy approach to reconstructing human subsistence over time. If you could go to the next slide, please. So just a little background on the region. We're talking about the Great Basin in the western part of the U.S., noted in the bottom right down there. So this is this hydrographic region. It's this closed hydrographic basin. It's a very large area. It's overall arid, very low precipitation, characterized by hot summers, cold, snowy winters. You can see a photo down there on the lower left of me taking some plant samples. And this is kind of the typical landscape here, is this sagebrush steppe. Sagebrush grassland steppe. Down in the valley bottoms in these kind of saline and alkaline soils, you have sagebrush greasewood communities. You get up in the mountains a little bit more, you get pinyon juniper woodlands on these mountain slopes. Next slide, please. So this area wasn't always like this. This wasn't always so dry and arid. This area was actually pretty wet at the end of the Pleistocene when humans first appeared in North America. This area has gone through pretty significant environmental change from this Pleistocene and into the Middle Holocene period and on through the Late Holocene. So it really provides some interesting context for understanding hunter-gatherer adaptations and how these are impacted by climate change over time. So just to briefly characterize the changes here, this was a much cooler, wetter area at the end of the Pleistocene. During the last glacial maximum, there was higher precipitation, and that resulted in the creation of these pluvial lakes. And you can see these outlined in the light blue in this figure right here. By about 14,000 years ago, this is the Bowling-Allerod period that Angie was talking about, these lake levels started dropping when we have remnant lakes and the emergence of these wetland systems in many of these valley bottoms. And this was, again, this is when we first started seeing evidence for folks coming into this area. This provided this very productive area for people to focus their subsistence on. These wetlands persisted into the early Holocene, even as the region began to dry. But then at the end of the early part of the Holocene and into the Middle Holocene, we see this increasingly arid climate emerge. These wetlands are drying up. By about 8,000 years ago, most of the wetlands and pluvial lakes are pretty much dried up in these valley bottoms. And so this is also true of the Chihuacan Basin, shown here on the left-hand side where the Paisley Caves are noted. This also underwent this transformation from wetland to kind of just a small, relatively small alkaline lake, saline lake in the valley bottom today. So this period of aridification sets the context for the discussion today. Next slide, please. Really interested in how hunter-gatherers adapted to this landscape. Also, this is adding to the environmental change during this time. There's the Mount Mazama eruption. So this occurred about 7,700 or 7,600 years ago. There's this tephra deposit across a large area of the western part of the North America. There's some evidence for a slight decline in temperatures, just a fraction of a degree Celsius for a few years. Overall, though, this seems to have localized impacts, less evidence for significant regional impacts, but there is some evidence that in the Paisley Caves region, at least, this led to a localized increase in the water level of lakes and marshes linked to this cooler period. So needless to say, this is a period of pretty significant significant climate change on the long term, and also from rapid events like this Mazama tephra deposition. Next slide, please. So again, this sets the context for understanding hunter-gatherer subsistence and land use during this time. And so this is kind of just a timeline of subsistence and land use in this northern part of the Great Basin. As in many parts of the North America, we see this gradual change from highly mobile, whether or not the folks in the Great Basin were large mammal focused or not is an outstanding question. That's part of what I'm going to talk about today. But this gradual increase in more sedentary lifestyle, broadening of the subsistence space over time. And so for this presentation today, there's an animation there, Steve, if you don't mind just hitting the next forward. So for the presentation today, I'm interested in this transition right here between this archaeological culture we know as the Western STEM tradition, which is kind of this first archaeological signature in this region, and this archaic period. And really getting at how meaningful this transition is in terms of subsistence and land use. We see, these are archaeologically defined cultures. We see transitions in the types of tools. Western STEM tradition toolkits are typically thought of as more hunting focused, evidence for, spear points used for hunting large mammals. In the archaic period, you see a proliferation of groundstone technology, increased use of basketry, although we do have some groundstone and basketry in the Western STEM tradition. But these are indication of this kind of changing subsistence regime. But really, I'm interested in how significant this change actually was. Do we have this radical transformation of land use associated with aridity, or are we saying just kind of a tweaking of an existing subsistence adaptation? So next slide, please. So just an overview of the Paisley Caves here. The Paisley Caves are a series of caves situated along this wave-cut shoreline, and this would have been cut into this landform by those pluvial lakes in their high stands or in the last glacial maximum. By the time people came here, the waters had receded. There's probably marshland just below the caves at that time. There's cultural deposits at the Paisley Caves, spanning the early part of, or the earliest evidence in the Great Basin for human occupation, about 14,500 years ago, all the way through the historic period. So this is really wonderful place to study human adaptation over time. In addition to that, it's this hyper-arid cave, so we have really great preservation in these caves. I'm going to focus my talk today. Our research broadly covers excavations in caves 1, 2, and 5 noted here. I'm going to talk about remains from caves 5 and caves 2 today in this presentation. Next slide, please. So this cave 2, the certificate of cave 2 is shown here. There's evidence for really intensive Western STEM tradition occupation here, marked notably by this well-preserved occupation layer down here at the bottom of the profile, this botanical lens, which has pronghorn hair, sagebrush bark, and all kinds of organic remains representing pretty intensive occupation spanning this Western STEM tradition. And then let me just back up, say too, in the stratigraphy here is again, there's excellent organic preservation. We have mostly windblown sediments, biogenic sediments. By that, I mostly mean bat poop, making up the stratigraphic matrix for many of these deposits. We also have tephra. This is that Mazama tephra that I talked about. And you can see it's a pretty substantial deposit. And that's that 7,000 or 600 or so year old marker right there. But when we have less intensive occupation in the middle Holocene following that deposition of the Mazama tephra. Next slide, please. So just a brief recap of the methods. I'm looking at coprolites spanning these occupations, and again, we have excellent preservation, so we have a really great coprolite record from this caves, and there's been a lot of work done on the coprolites from Paisley Caves over many years. Most interested in this project, we're mostly interested in trying to reconstruct dietary decisions on a short-term, fine-grained scale, and coprolites, of course, are ideal for that. So our project comprises fecal biomarker analysis, looking at sterols and bile acids to determine whether these samples are human, and then also kind of the classic traditional multi-proxy analysis, looking at macrofossils and microfossils to analyze diets. Next slide, please. So from this Middle Holocene context, there are 15 coprolites that we've identified from this context. 5 of these have been radiocarbon dated. Here's a nice gallery of some of the samples that we're working with. And this is prior to me taking my subsamples out of them. Some of these have been radiocarbon dated to this Middle Holocene period. The rest are stratigraphically dated. And we are working on getting radiocarbon dates on the remaining samples to more securely place them in time. Next slide, please. I'm going to focus in on just 9 of the samples of these 15 that have the best dietary evidence for this presentation today. And so the first and most important thing for our study is establishing which of these coprolites is human. So this is a long-standing issue in coprolite research, trying to determine with any certainty whether feces left at a site were deposited by a human. You can look at morphology and inclusions, but these can be misleading. Particularly with our research, we're finding that a lot of coprolites may look like a carnivore coprolite because they have a lot of, for example, rodent bones or fur visibly in them. Actually, are human. Human diets are overlapping with carnivore diets in a lot of situations at these caves. And so we're we're looking at biomarkers in these coprolites. These are these complex organic compounds. We're looking at a combination of 5-beta stanols and bile acid biomarkers to try to identify coprolites as human. So copper stanol is the major 5-beta stanol in human feces, and this is produced as cholesterol, is converted to copper stanol in the human gut. And then human feces also have high levels of deoxycholic and lithocholic bile acids. And so we analyze these fractions of the coprolites using gas chromatography, mass spectrometry. It produces this chromatogram like this, and we can see, for example, in this sample right here, we see a high peak of copper stanol, and that in tandem with peaks of deoxycholic and lithocholic bile acids indicates that these are human. And so you can read more about this, the methods in this paper that our team published last year, cited down here at the bottom. Next slide, please. So we've determined that all 15 of these samples are human, which again, it's been kind of this long-standing issue in coprolite research is how do you determine whether, with any certainty, whether you're actually looking at a human sample? So just going through some of the results of the analysis, here are some of the microfossils, or macrofossils, sorry, from these coprolites. Several samples have typhus seeds, so this are cattail seeds. We have mentzelia or blazing star seeds in several samples, juncus rush seeds, atriplex saltbush seeds, scirpus, bulrush seeds. We also have evidence for consumption of mammals, in this case, small and very small mammals. I only have faunal analysis data on 2 of these samples, but even just those 2 samples so far has some really interesting results. So in this sample, number 170, we have Paramyscus, which is a deer mouse. Not pictured, we also, in another sample, we have evidence for consumption of montane vole, kangaroo mouse, and hare jackrabbit. And that's all in the same coprolite. We have bones from all 3 of those organisms in that coprolite. Next slide, please. So people are consuming a really diverse diet. You can see here, these are some of the microfossils. Again, we just have the faunal analysis completed so far on 2 of these samples, but both of them have a similar signature. And then I just noted here fish scale in one of these samples. So this is the range of different types of macrofossils, focusing on seeds primarily here. Next slide, please. With pollen, and this builds off of what Vaughn was saying, and you know, we need to have a criteria for indicating intentional consumption in coprolites, because obviously pollen is in the air we breathe around us, it's in the water we drink, there's a lot of ambient pollen rain. So we have these criteria that we use to try to establish intentional consumption of plant material. And again, we're looking for aggregate grains with the idea that these aggregate grains are going to be adhering to the plant that produced them, even if we're not talking about consumption of actual flowers and anthers. We're assuming that aggregate grains are more likely going to be on the vegetation from the plant that produced that pollen. Also high frequency and or concentration. And again, this builds off of work that Kristen and Carl and Vaughn also did many years ago. And so here are some of the criteria. So in this slide here, I'm showing some of the highest confidence of consumption. So we have, in one of the samples, we have, they're pretty crumpled, but what looks to be a type of Fabaceae aggregates. Again, this is these are mostly insect pollinated taxa. So this is a very high percentage in this sample. And Asteraceae, highest finding, mostly insect insect pollinated, and we have aggregates and high percentage. For these wind pollinated types down here, we do have in sample 2 of these samples, we have a lot of aggregate grains of grass, Poaceae. You can see this large aggregate right here, and again, higher percentage in these samples. And then we have quinoam pollen aggregates, and then also high percentage in this sample right here. So this kind of gives some additional dietary information that we couldn't glean from the macrofossils. Next slide, please. Also looking at phytoliths from these samples. And so the phytoliths, again, give us just another window into some of the different components of the diet, the types of plants, but more particularly the plant parts that were consumed. And so for these samples, the most important or most novel observations that we have is that there was several types of monocot morphotypes and monocot epidermal sheets in many of these samples, and suggesting that folks were eating monocot vegetation, mostly epidermis, so leafy green type of material. Next slide, please. And so kind of combining the pollen and phytoliths together into one table right here. This is some of the insights that we had of dietary contributions represented these coprolites. And interestingly, only in these two highlighted in green here are actually linked up with the macrofossils. So the rest of this is, again, just novel insight from that kind of shows the strength of this multi-proxy approach to reconstructing diets using these using pollen, macrofossils, and then also microfossils. Next slide, please. And so this is still very much a work in progress, and I'm currently writing this up. And you know, I'm working on a more in-depth comparison. But just to kind of go back to this question that I posed at the beginning is how different are these lifeways between this Western stem period and this archaic period? This kind of this ongoing debate of whether this is Western stem folks had a Paleo-Indian adaptation or what we might call Paleo-Archaic, kind of just an early pre-archaic kind of adaptation. And we do see continuity in a lot of different types of foods represented in the coprolites. Again, this Western stem data is published in this paper down here that we published in Archaeological and Anthropological Sciences last year. We see a lot of continuity in things like cattail, amaranth, grass, legumes. Wetland taxa are still important from the Western stem to the archaic. Folks are still eating things like hare, jackrabbit, rodents, and through both periods, we see people focusing on subsistence resources that offer a pretty low return for time invested in procurement. And so, again, when comparing these two subsistence strategies between the two, it does seem like we have something more like what Dave Madsen has talked about, where we have this less of a radical transformation of lifeways and more of just a change in the scale of this already existing broad-based subsistence strategy, and probably in response to the disappearance of some of these more productive wetland areas and emergence of this kind of arid environment. And so moving forward, we're exploring the biomolecular analysis as a means of identifying additional plant foods. So looking for different types of biomolecules associated with certain plants and animals in the coprolites. And again, that's going to give us another facet of information to use for a multi-proxy approach. And so that's all I have for you today. I know I've gone over my time. I just want to say one last thing. When I came to graduate school, next slide, please. When I came to graduate school, I was interested in rocks and stone tools, and I started taking classes with Vaughn, and it was through Vaughn that I learned the kind of this whole new world of using plants, macro and microfossils for understanding past human behavior. And if it wasn't for what I've learned in Vaughn's lab, I wouldn't have had the opportunities that I've had in my career. And so I just want to say thank you, Vaughn.
Yeah, great. Thanks, John. Yeah, I think Vaughn had a lot of those sort of influences, whether you came to study nautical archaeology with George Bass or some of the later professors there, or came to study stone tools with geoarchaeology with Mike Waters. You know, if you ended up taking one of Vaughn's classes, you could see the you know, some of the micro fossil and even some of the molecular levels that he didn't himself do, but he introduced people to. If we have time for questions, if there are any, if not, we'll keep chugging along. We are running a little bit behind here. And I'd like to introduce Rosanna.
Questions coming through, so maybe we can move on. There's no questions coming through.
Great. That's probably for the best. And you know, we can always have time. We have everybody's emails these days, so there's ways to do that. So next I want to introduce Rosanna Paredes, and she's going to talk about Vaughn Bryant's influence on Peruvian palynology.
Hi. Well, I think I have the same situation as John. So I'm trying to access the security and privacy here.
Okay, I think we can, if necessary, we can do the same thing we did with John, where you email it over to Steve, and we can bop down to the next one and come back to you in 15 minutes or so. Does that sound reasonable?
Yes, thank you.
Yep. Okay. So for those of you joining us, we're going a little bit off of schedule here, having some technical difficulties, but we're going to come back to Rosanna's talk on Vaughn's influence on Peruvian palynology. And if Jennifer O'Keefe is ready, we're going to be talking about medicinal palynology of honey produced adjacent to a South Carolina biodiversity hotspot. All right, we see you, but we don't hear you. There should be a little strip control.
There we go. How's that?
Perfect.
Let's
Yep.
see.
Your screen. Perfect.
You all see my presentation?
Absolutely.
Or do you see my presenter screen?
We see your presentation.
Excellent. Okay. So bit off piece, jumping ahead to melissopalenology. So I have been doing melissopalenology with Vaughn for a very, very, very long time. It's been an interesting process. We know that in the United States, melissopalenology has a very long but sparse history. We're really lagging behind the rest of the developed world. You know, we had our first melissopalenological study in the US back in 1908 with Young's analyses of hundreds of honeys that had been displayed at the 1903 World Fair. And then not much happened until the 1930s with Pellet's American Honey Plants, which had a section on pollen and honey, closely followed by the 1930 Iowa Honey Plants, which had another section on pollen and honey. That was by Pamela, I think. Read that years and years and years ago. My great-grandmother had a pamphlet of it sitting at her house. And then a bunch of minor reports through the 1960s and 70s, although standing out among those was Wenzel and Todd's 1942 paper on U.S. honey composition. Melissopalenology in the U.S. really began in the 1970s with Meredith Liu's work on Louisiana honeys, quickly followed by Vaughn getting an interesting call and letter from USDA saying, hey, we've been contacting palynologists all over the US looking for someone to analyze honey. We think we've got adulterated honey. Can you do it? Vaughn always said, he said, sure, as he's looking at the dollar signs going, money for the lab, excellent. He also said that was probably one of the best and the worst decisions he ever made. The summer of honey, the first summer of honey was hell. He lived at the microscope. Carol was not thrilled with the amount of time at the microscope, but he learned a lot. And through his tireless efforts between 1975 and 2018, he finally got people to realize that we needed to think about truth in labeling about honey in the United States. And in 2018, the FDA published its first truth in labeling guidelines for honey. And melissopalenology has begun to explode. Unfortunately, it began to explode just as Vaughn began to implode. At the moment, following Vaughn's legacy, there are 5 of us in the US who are self-identifying as melissopalenologists. John Jones, Christy Briles, and I studied with Vaughn. Sophie Warney at LSU worked extensively with Vaughn in the latter years of his life. And Irina DeLucina has had her work vetted by Vaughn and made visits to Vaughn's lab several times. In fact, we overlapped on one of her visits. It was kind of fun. If there are more of you doing melissopalenology, please, please, please let me know. Bee Culture magazine is really campaigning for an article retrospective of Vaughn's life with a listing and map like this one of people doing melissopalenology in the US. And honestly, 5 of us is a very slender thread to hang the science on. With the increased emphasis on truth in labeling. So if beekeepers want to label their honey with type in the US, they now have to have an analysis to back that up. It means demand for melissopalenology is really increasing. And honestly, the work is heavy. I think none of us realized just how much Vaughn was doing. Today, though, I want to tell you about a project Vaughn insisted I take on near the end of his life, I got one of those infamous 32-point font, boldface, hot pink emails from Vaughn about 18 months ago that said, Jen, you should take on this project. It looks like a lot of fun. Quickly followed by an email to David McFawn saying, hey, David, my health isn't so great. I suggest you contact Jen O'Keefe. Should be thrilled to take the project. Thanks, Vaughn. Interestingly enough, I was thrilled, and it has been a lot of fun, although quite a lot of work around the rest of my job. So the Congaree National Park is located in the larger Congaree International Biosphere Reserve. A bee yard's located on the bluff directly adjacent to the Biosphere Reserve, or adjacent to the national park. You can see the swamp tupelo groves from the bee yard. From this location, the bees are foraging entirely within the International Biosphere Reserve and crossing the river into the national park. So we're looking at bee foraging habits in an internationally protected area with incredibly high biodiversity. Of major concern to David and regional beekeepers is the observed disjunct between published honey plant lists and what they're observing on the ground when the bees are foraging, and the feedback they were getting from melissopollinological analyses that Vaughn had been doing for them for many years. It was clear to David that analyses of single annual honey samples was not giving him enough information, that something was going on, and he'd been talking to Vaughn for a while about doing a large-scale project looking at honey samples over the course of a year to track foraging behaviors. Vaughn had been doing this with various students, including Pierre Lau at Texas A&M for a long time, and it was in a way sad that he passed it off to me and didn't get to do it himself, because it's been an interesting project. David was able to secure funding to do detailed sampling through 2020. And we set up a pilot program where David was collecting 2-inch squares of comb from the end of February through the end of November 2020, when there was new honey, every week, and sending those squares of comb in urinalysis cups to me in Moorhead, so they're kept sterile, they're kept cool, and shipped through the mail. David had thought the spring nectar flow in the area was about from the 1st of April through the 1st of June, with a bigger autumnal nectar flow. He'd always seen lots of autumn nectar. So we were able to get honey samples from the supers. He didn't want to touch the brood boxes for this study. So he's looking specifically at new capped comb in the supers above the brood box, and was able to take samples March 28th, April 4th, 9th, 18th, and 24th, May 1st, 8th, and 15th, June 3rd, 12th, and 28th, and October 29th, 2020. The rest of the year, there was no new honey being produced in the supers. From looking at his hives, David knew that the bees were collecting nectar and pollen during the rest of the year, but they were not putting it in new comb above the brood boxes. He suspects everything was going into the brood boxes themselves from mid-March, and we're going to take a look at that in the next study. The autumn nectar flow in 2020 was really not very strong there. You got about half a frame of honey, which was a little worrying. And during the summer, the nectar dearth was much longer than he'd previously observed. So once we got this stuff back to my lab, we processed it in partly the typical method and partly our in-house method. We're not set up for acetylysis with our undergraduate-driven lab. Instead, we use the Scholes et al.'s cellulase and pectinase enzymatic method. With honey, we've had to adapt this a little bit, especially with wax-rich honeys. We hit them with a little bit of potassium hydroxide for no more than 3 minutes, that we don't damage the
outer surface of the grains. You know, too much potassium hydroxide on fresh pollen is not a good idea. It gets very glassy. We end up with mounts that look virtually identical to 3-minute acetolysis rather than the 9-minute. They're pale. We have to stain them with saffron. They're not the nice rich brown Vaughn always made, but it works. Our analyses are fairly similar to Vaughn's, although I will point out I count with a self-calculating rarefaction curve rather than just counting 200 non-tracer grains. We found that with extremely diverse honey samples, we really do need to count a lot more. And it's better to count until this rarefaction curve reaches its asymptote than to do an artificial cutoff. Looking at our data, we were able to pick out some trends. First of all, there's a lot of variation in concentration value in these honeys, but they're typically quite low. If we look at the honey concentration category, most of these are type 1 honeys. We had some type 2 honeys in April. I'd like to point out that the June 3rd honey was a type 4, as was the October 10th honey. We'll come back to this October 10th honey in a few minutes. The June honey was the most pollen-rich honey I've ever seen from all of the samples I've looked at from South Carolina. It was rather impressive. We did have 3 monofloral honeys in this. These are honeys with more than 45% of a single dominant pollen type in the honey. The April 18th sample was dominated by holly pollen. The April 24th was dominated by sumac pollen, the June 3rd by wild mustard family pollen, and all 3 contain multiple seasonal supporting pollen types in addition to the predominant type. The remainder of the samples had very mixed floral sources. Those floral sources did, however, provide information about when various pollen types were and were not present in the samples. We were lucky enough to have a fairly substantial database from NASA and also from Clemson's Agricultural Extension Agency showing purported bloom times of various bee important plants, especially those that are known nectar sources. And we had assumed, and David had assumed, these lists would give us a great idea of what we would see in the honey. And in fact, they really didn't. Of the forecast types, only 24 occurred in the Congaree samples. There were an additional 28 types, 2 of which were indeterminate, documented in the samples. The predicted samples are here in yellow and green. The things that showed up in the honey are the black lines showing which months, and in some cases parts of months, the sample showed contain these pollen types. One of the things that stands out here is we're seeing a lot of variation in bloom time, and the bloom times are not necessarily matching the published bloom times for this area. Most of these bloom times were documented in the 1960s and 70s, and it's become very clear that these are changing and may not be accurate through the whole region. We're seeing some pollen occurrences like chickweed, alder, dandelion, and corn, and tick seed appearing in samples for much shorter periods of time than anticipated, while others like tupelo, tulip poplar, and holly are present for much longer than anticipated. Of interest and quite a bit of concern to David and other beekeepers in the South Carolina Beekeepers Association is that goldenrod pollen, typically a major source of pollen and nectar in the fall, appears to have had a very short early bloom time in 2020. And just from observing the plants, David said he saw similar things this year. The goldenrod bloomed back in June and is not in bloom now. So one of the things David was able to determine is that nectar flow started much sooner than anticipated in the Congaree River area. Nyssa, Salix, and Cephalanthus, the Tupelo willow and buttonbush all bloom much earlier than predicted. Holly bloomed in mid-April through the 1st of May, with tulip poplar blooming the first half of May, which is a little bit earlier than anticipated. We also saw privet, which is a typical summer bloomer through much of the Mid-South in the first half of May in this area. The mustard family plants, the small Brassicaceae, bloom mainly in the first part of June. The autumn sample was very interesting, and we did just have the one autumn sample. This sample included Artemisia, our mugwort sage, nettles, urtica, and mulberry, our ribes pollen. It did not contain any goldenrod. Okay? It didn't contain many of the other typical fall blooming taxa. No fall Asteraceae that we typically find in the region, like the ragweed, like the Joe Pye weed, like our goldenrod, and so on. It was very, very strange. In fact, when I stepped back and compared it to the rest of the samples, it was virtually identical to that type 4 honey we saw back in June. David got to thinking about it, and he strongly suspects the bees weren't foraging on honey plants. They had found an abandoned hive and were robbing away to fill their comb. So we're going to keep a closer look on this. Overall, we saw 62 pollen types in the honey over the year period, 38 of which had not been previously documented for the region. As we move forward, a statewide study expanding this to each USDA extension region in the state has been submitted with the South Carolina USDA block grant. We've been recommended and are answering questions, so hopefully we'll be funded and be starting a big statewide project in January and February. David and the other beekeepers are highly concerned about the published plantlets and their accuracy statewide after this short study, as well as realizing that the pollen data seems to be backing up their observations that bloom times are changing statewide. And they want to know how they're changing statewide. So hopefully we'll have a one-year study followed by a longer study to really put this in context. In this first study, we only looked at surplus honey, the honey that was being stored above the brood boxes in the supers. Next time, if there's no surplus honey, we're considering looking at brood honey, provided it can be sampled without getting into the brood cells. We're still going through the possibilities with the other beekeepers who've signed on to the project, and we'll keep you posted on that. So special thanks for this project go to the South Carolina Beekeepers Association for funding this work, and Sydney Brady, who provided the bee yard location for the original study. And always thanks to Vaughn, because we wouldn't be doing any of this without his efforts. Thank you all.
Well, great. Thanks, Jen. As a former amateur beekeeper myself, I always find these interesting, although I never got quite into the melissopellinology side of things. Maybe one of these days. With that, I think we're gonna, for those of you who might just be joining us, we're a little bit off schedule with some technical difficulties, and we're a little bit behind schedule. We're gonna be going back to Rosanna Paredes, Vaughn Bryant's influence on Peruvian palynology talk, I believe.
Hi.
Great, we can hear you and we can see your screen.
Yeah, I figured it out. Okay. Let's see. So everything is fine? I mean, you can see my screen and also hear me?
We can hear you and we can see your screen. It's not in presentation mode yet.
Oh, okay.
Yep.
It is now?
No, it's still the working PowerPoint. I think if you just hit the slideshow button and hit first slide, it should pop into it.
I already did it. Maybe it's the internet connection.
No, I mean, we can see your screen. It's just the sort of where you work in the PowerPoint as opposed to the slideshow presentation. If that makes sense.
But I already, yeah, but I already changed into a presentation mode, maybe.
Okay. I mean, we can see your slides, you know, so I think it'll be fine even if we just scroll down. So.
Okay, yeah. Okay, well, I'm going to start then. Thanks for the invitation. I'm happy to be here with you all. Yes, and I'm going to talk about Dr. Bryant's influence on Peruvian palynology. This is actually the first time that I am talking about this, and I thought it was important because we know that he had a huge influence in the US, but also around the world, right? So I made this timeline. I don't know if you can see it, about palynological history in Peru, and this is the time around the late 90s, yeah, when Dr. Ryan started to provide us publications and all his research work on pollen. And before that, we only have a few documents on palynology in Peru, and they were related basically to the oil industry. Palynology was not a discipline per se, but after we received his influence or started to receiving his influence on academic research, we improved in palynological work. As you can see here, we are still working on that. Actually, Peru is one of the countries in South America that there are not many palynologists there, but it is still developing. In Peru, these are the main areas where we conduct palynology. Archaeological palynology and paleoethnobotany are the most predominant ones, and now we have started to contribute to melissopalynology, and this year we have been working on a line or a proposal to test if forensic palynology will be viable to be conducted here in Peru. I bet most of you have heard about the story regarding the Piquimachay site in Ayacucho. Dr. Ryan was called by Dr. McNeish to continue Dr. Collins' work on coprolites in the summer of 1971. And I remember every time he started to tell us about this story or this experience in Ayacucho, he mentioned that the flight from Lima to Ayacucho was very scary and bumpy. He was able to see the oil leaking through the window. I bet most of you have heard about this. And to me, it was the first time when I started my grad students to listen about this situation. I didn't know that before getting to Ayacucho was so complicated. But yeah, this is actually one of his first works in Peru, and he started with a coprolite investigation. Then, well, here you can see a picture from his Ryan and Dean 2006 publication, where he's in Dr. Collins' bedroom and laboratory. You can see all the samples and the materials that he was using, and Dr. Ryan continued to look at. Throughout the years, we have been receiving his influence by all the publications and all the work that he conducted on palynology. Here are some of the main articles. Of course, there are more. And archaeological palynology is the most important subdiscipline that we are working on in Peru. He has influenced the Andean region and across the coast also. We differentiate across in the north, central, and south areas because of the culture that's settled there and the people and the archaeologists that work there. They are different groups and they are well known. So all of these specialists use his publications, his investigations as a reference for field and laboratory work. And of course, he helped everyone with pollen identifications. Okay, I think that the most remarkable or the huge contribution that he made to us is that he trained the first palynologist, Peruvian palynologist, Dr. Luis Guaman, that is here in the picture. He, after finished his studies in biology, was interested in palynology, but at that time, there was no one doing palynology in Peru. So he needed to look for an advisor. He started to email many specialists from Europe and from the US. And as always, Dr. Ryan was the one that replied immediately. So he talked to him, shared his interest, and then he traveled to Texas to start an internship in the TAMU palynology lab. After obtaining that experience on processing, identification, and how to conduct these palynological investigations, he returned to Peru to start working on the PAN project, which was a huge archaeological project in northern Peru. And they were including the analysis of pollen from soil samples, from ceramics, and different type of samples. But they realized that there was no pollen lab, a place to analyze these samples. They needed to send the samples to the US and then process them there, and it was very expensive for the project. So they decided to build the first pollen lab in Peru. And guess what? Dr. Ryan helped Luis, to set the lab up. He sent lots of books, helping with looking for tools, guidance on what to buy, how to install the equipment, etc. So this laboratory, the Palynology and Paleoethnobotany Laboratory, is located in Universidad Peruana Cayetano Heredia in Lima. We do diverse areas of investigations, specifically archaeological palynology and some melissopalynology. Throughout the years, we were able to develop a Peruvian pollen reference collection, and we are now working on a pollen atlas that there are some details that we need to finish, and we hope that we publish this soon, because there is no a specific or a particular pollen atlas from Peru. We don't have any, so we want to publish this as soon as possible. And of course, we have a good pollen reference from Latin America. And there is an herbarium associated to the lab, where we keep the modern samples. Okay, you can see here is part of the reference. In the lab, you can find specialists, students, interns, they work together. There have been some publications, they do presentations, and there are theses and dissertations from these students that have been developing this line in Peru. Okay, this laboratory is located in Lima, but we also do collaborations with specialists from other regions of Peru, as this is the only laboratory, as I've mentioned. In the coast, people mostly do archaeology and melissopalynology. In the Andes, just archaeology, and in the Amazon, we are starting to do melissopalynology. This year, a student made this proposal on the use of palynology in forensics and how viable will be this to be part of the laboratory as a specific line. So we are testing that. We hope that it will hold the attention of the specialists here in Peru. Okay, so to continue with this training, Lucho sent one of his students, myself, to continue learning on palynology. I was very interested when I finished my third, 4th year in my undergrad studies, but I wanted to do archaeological palynology. So I talked to Bon, and we started working on the relationships between plants and peoples in northern Peru, third time, from the Lake Archaic until today. So I try to combine palynology, paleoethnobotany, TK, the study of TK, and different approaches. But when I finished my dissertation research, I needed to stay an extra year in the US because of legal issues. I was an alien, so I could not graduate if I wasn't there for another year. And I didn't know what to do, so Bon convinced me to do some melissopolynology. At that moment, we realized that there was no information outside Peru on Melisso. So I got very interested, especially that since I was a kid, I always heard that honey from the Amazon, the Peruvian Amazon, was better than the ones from the other regions. People associate this honey with medicinal properties. Of course, this is an oral knowledge, and there was no proof, or at least a pilot study to guide us and try to test this, right? So we collect some samples from the Peruvian Amazon, from the highlands and the lowlands, and got this interested study. This is a pilot one, but we got, well, you can see here some of the pollen grains, and most of the plants that were identified from the honeys were associated to medicinal plants. They had medicinal properties. So we proposed that to know if these plants have a benefit in your health, first you need to know what is in there, right? In the honey. So the botanical content, the pollen content, and then it will be great to continue the study with phytochemical analysis, biochemical analysis, to actually know which chemical compounds are the ones that are giving these properties to the honey. So this is the first melissopolynological study that was published in an international journal regarding Peruvian samples and from the Amazon. There is no information on pollen grains available outside Peru on Amazonian honey. So we wanted to do, or to start doing that basis, right? And I'm happy to mention that, well, when I finished my grad studies, I returned home because of COVID. Many plans were canceled, right? But a few weeks ago, we were able to return to the lab, and we just start, I think, the most beautiful project I'm involved with, the Von Bryant Honey Project. This is how I call it. And we are analyzing honey from stingle bees from Peru. These are native bees, and we don't have any information about them. We know, because of the reference from other countries, that the honey produced by stingle bees have more medicinal properties. They are better in health for us than the honey produced by apis or other bees. So we wanted to know if this is also happening here in Peru. And we are trying to give an ethnobiological focus to the project, combining palynological, entomological methodologies, and also studying the TK associated to the production of this honey and its medical uses and properties provided by the local communities. We are working with the Awanjun people, which are located in the Amazon, northern area of the Amazon. And these are the ones that work with stingless bee honey in this region. They keep the bees and help in the production of honey, and they sell it regionally to people. It is not more expensive than the ones produced by apis, but I heard that it is better. So we are going to test that or start testing this oral knowledge. One of the main objects of this study or this project is to start developing this database from the Amazon, the pollen from the Amazon, the Peruvian one, because there is information from the Brazilian Amazon, Colombian Amazon, that we used to identify the pollen grains in the previous study, but there is none on the Peruvian one, just the ones we identified with Dr. Brian in the pilot study. So we want to start developing this and contribute to melisopollinological research in Peru by educating the beekeepers, the customers. People will be willing to pay more or to buy expensive honey because it is certified scientifically that its content will be better for our health. So this will be also better for our economy. And of course, we want to recognize the cultural identification or the cultural knowledge, the cultural value that is behind this production of honey from our native stingle bees. So as a conclusion, Bond definitely contributed to the development of palynology in Peru. These are all the ways that we were receiving his knowledge, mentorship through fieldwork, advising, publications, pollen identifications, and everything you can see here, and etc. As most of us, he made this influence in our professional and personal lives. So that's everything that I have.
Great, thanks, Rosanna. I know Vaughn would be thrilled to see you continuing not just with the archaeological palynology, but expanding your melissopaleontology down there. He always, as you pointed out, was very generous with his time and resources, both within his graduate students as well as people from around the world who contacted him. And that's a great way to turn it over now to Kristen Selbulik, who's going to be giving our keynote here. Vaughn M. Bryant, Words of Wisdom, Lessons Learned in a Life Well Lived. And I know we're all looking forward to this one.
So I'm hoping, can everybody hear me?
Yes, we can hear you.
Great. How about seeing my screen?
Not yet.
Okay. So Tim, where do I see the change presenter stuff?
Right.
Yeah.
It shows me you have control. Kim or Steve might be able to address this a little bit better. I actually haven't had to share my screen, but I think it's in the toolbar there.
Okay.
Kristen, you should see a little box pops up that asks you to share your screen.
Yep, sharing, it says. How about change presenter?
My little cheat sheet shows that you are currently the presenter.
Okay, audio, webcam, handouts. It's on the left.
You should have got a box that popped up asking you to share your screen.
Like a standalone pop-up window.
Yeah.
Okay.
Okay, I've just taken you off presenter and put you back on, so you should see that box requesting to share your screen.
Yeah, location, full desk, screen recording.
I wonder if the box is hidden behind another window.
No,
Maybe.
I think you're on that where it says full screen recording. That just is choosing which screen you're going to share. I think that's the drop down for which screen.
Still looking.
Okay, we'll try changing the presenter again. Just a second.
Okay.
And we'll...
And there'll be a box that comes up.
Yeah.
It says go to webinar, would like to capture this computer screen. It says deny or open system preferences.
That sounds like the security issues that some of the other presenters have had as well. That's nothing to do with us, I don't think.
All right, I'm opening the password. Could I send this to somebody?
That is something we've done. We've been sending them to Steve.
Yeah.
And he can advance slide for you if that's easier.
Okay, thank you. Sorry about this, everybody.
That's okay. You're not the first.
I'm sure.
Yeah, the blessings and the curses of these various internet platforms. They're all a little different, and they all interact a little differently with systems, and perhaps your institution's IT. Who knows? So.
Okay, Steve, I just sent you something. The title of it is Student and Early Career Event. I just replied to that.
Oh, great.
Okay, I will have that with you as soon as it pings through to me.
So how long does that
file size?
Cross the ocean?
Yeah, we always think of it as instantaneous, but it's not quite really. So.
And of course, we're having road work out here. I can get. I'll start with an intro, Steve.
Well, if you want to, we could actually take the break now for 10-15 minutes, sort this out, and then do it that way. Do you think that'll be best way of doing it? Then we're not.
That's fine. That's fine as well.
Yeah. Okay then. So we'll all take a break now and reconvene in 15 minutes.
Okay, so right at the 30-minute mark of everybody's hour.
Yeah.
Okay.
Yeah. So at
sounds good.
17:30 BST, and then wherever you are in the world. Okay. So thank you very much, and we'll try and sort this out.
Should I send this to Callum Cleary as well?
Not to Callum, no. If you could send it to me and it might be best to send to the meeting email, which I think is, I'm trying to remember what the actual email address is, AASP-meetings@gmail.com.
Okay, thank you.
Well, thanks everybody for your patience. As Steve said, we're going to take our break early, or early in the schedule, perhaps probably late in our actual timed session. And we'll be back at the 30-minute mark of the hour for the keynote on Vaughn. Thank you.
Hi, Kristen. Are you there?
I am.
Yeah, this is Kimberly Bell, one of the organizers. One thing that some presenters have had luck with is if they log back out of the go-to platform and then log back in, sometimes they're able to get by that security issue when they come back in. So if you wanted to try that, we could give it a shot and see if we can get your screen up.
Okay, I'll do that, and hopefully you received the copy as well. I came on, and of course I was listening to the conference all yesterday and making sure I could get on, I could do the audio. I just wasn't able to test the screen share.
Yeah, and Monday and Tuesday we had no troubles at all with the screen share, but today it seems there's been a few problems. So yeah, why don't you just try logging back out and coming back on? That would be the best case scenario, because then you have, if we can get you to work, you can have control over your own slides. But if not, then we'll get it up for you.
Yeah, that's okay. I think, sending it to you too, I don't have that many slides, but they have a lot of pictures on each slide, and I think they'll be very helpful.
Okay.
But yeah, having me have control of the slides isn't as imperative, I would say.
Okay.
So I was on, I think it was yesterday, and things, I think, in general have been going well. How about from your perspective?
Yeah, I mean, from a technical perspective, things were really quite smooth the first 2 days. Just a few hiccups today.
Mm-hmm. Yeah, sorry about that.
Oh, no problem. These things happen.
Where are you physically at?
I'm in Calgary, Canada.
Okay. They, of course, too, decided to take out the entire road in front of my office today, of all days. So there's a lot of noise here as well.
Of course, it's always on the most inconvenient day. I'm just gonna pop out to have a quick break here, but we will see you shortly.
All right, thank you.